Mortality statistics from the Center for Disease Control and Prevention show cancer has been the second leading cause of death and accounted for 22.5% of all deaths that year. Cancers often become remarkably lethal by developing multi-drug resistance, or MDR, making treatment very difficult. One well-studied protein identified to play a major role in multi-drug resistance is P-glycoprotein-1, abbreviated Pgp. P-glycoprotein-1 belongs to the superfamily of ATP-binding cassette transporters, or ABC transporters. These use ATP to actively pump molecules in one direction across lipid membranes. P-glycoprotein-1 is often overexpressed in cancer cells and contributes to multi-drug resistance by pumping chemotherapy drugs out of the cell. The crystal structures of ATP-binding cassette transporters and P-glycoprotein-1 used in this video are not human, but are identical enough in sequence and function to human receptors to make valid comparisons. P-glycoprotein consists of two similarly structured subunits. In this diagram, one subunit is colored red and the other is colored white. Each subunit has a cluster of 6 transmembrane domains, or TMDs, connected to a nucleotide binding domain, or NBD, which is suspended down here in the cytoplasm. Transmembrane domains from both subunits partially wrap around each other like a handshake. The nucleotide binding domain binds ATP to power the transporter. In the unbound state shown here, the receptor's mouth opens into the cytoplasm with nucleotide binding domains 30 angstroms apart. Substrates bind between the transmembrane domains in a 6,000 angstrom cube pocket. P-glycoprotein-1 can transport molecules ranging from 330 to 4,000 Daltons in size. It has very broad selectivity for hydrophobic substrates and more limited selectivity for hydrophilic substrates. These crystal structures show how P-glycoprotein-1 transports substrates out of the cell. First, the substrate must bind to the exporter. Substrates can bind by entering the mouth of P-glycoprotein from the cytoplasm. However, many hydrophobic substrates can bind from inside the lipid membrane by slipping between the two subunits at the edges of P-glycoprotein's mouth. These hydrophobic substrates, including chemotherapy drugs, paclitaxel, and actinomycin D, have substantially higher affinity for P-glycoprotein-1 when absorbed from the inside of the lipid membrane versus from the cytoplasm. This is likely due to hydrophobic substrates being more concentrated inside the hydrophobic lipid bilayer than in the hydrophilic cytoplasm. Binding of substrate causes a small conformational change, allowing ATP to bind the nucleotide binding domains. The nucleotide binding domains dimerize in a head-to-tail manner, with a twofold rotational symmetry to form 2 ATP binding sites. Free energy of ATP binding and nucleotide binding domain dimerization is used to invert the transmembrane domain clusters and open away from the cell. The exact opening mechanism involves a rotation and rearrangement of transmembrane domains from both subunits. The inversion causes P-glycoprotein to lose affinity for the substrate, and the substrate quickly diffuses away. Then, one of the ATP molecules hydrolyzes to ADP and the nucleotide binding domain dimer dissociates. This returns P-glycoprotein to its unbound conformation, ready to transport another substrate. Researchers want to study the P-glycoprotein structure and mechanism to develop inhibitors. Inhibiting the P-glycoprotein in cancer could allow chemotherapy to work effectively for multi-drug resistant cancers. Today, there are P-glycoprotein inhibitor candidates that allow chemotherapy to kill multi-drug resistant cancers in vitro. A few have even made it to phase 2 and 3 clinical trials. However, none have proven useful enough for widespread use in humans. The problem of multi-drug resistant cancer is far from being solved. The discovery of a very effective P-glycoprotein inhibitor safe enough for human use would be a major leap forward for treating multi-drug resistant cancers that claim numerous lives today.